Okay, 3, 2, 1, it's jam.
What's up, science fam? Yeah, cells are tiny, but what exactly are they themselves made of? What is the fundamental unit of life made of? What we're going to talk about this session is the biomolecules. So these are the molecules of life, and life technically is just space dust that started thinking about itself. But what exactly is this space dust? So what are we up to in this session? We're going to discuss the significance of water to life, explain how carbon is the backbone of life, and categorize different biological molecules according to their structure and function. So what do all forms of life need? What molecule is absolutely necessary? It's water. It's known as the biological medium. We have here a kingfisher bird that is, fishing for fish. So we have one organism that's habitat, normal living habitat is water, and another that has to go into water in order to get its food source, as well as, it uses water as a source of hydration. So why is water so important for life? It's because of its different properties. So first property is water's structure. It is a molecule that has polar covalent bonds, and it's highly polar. So what happens is that oxygen is highly electronegative, meaning it likes to hog all the electrons, creating strong partial charges. So oxygen is partially negative, and the hydrogen atoms are partially positive. This makes it so that the unique properties of water come directly from its structure. So that structure results in 4 emergent properties. So emergent properties are a thing that happens in life. It's a unifying theme of life. The more complicated something gets, the more properties emerge. So these are the properties that emerge at the level of the molecule in water. So property number 1, cohesion and adhesion. So these are different from each other, and they're the result of what we call hydrogen bonding. In your chemistry class, hydrogen bonding is an intermolecular force of attraction where the partial charges between hydrogen and other highly electronegative molecules make them have a strong bond between molecules. So this results in cohesion, which holds water molecules together, and adhesion, which lets water cling to most surfaces. That's why when you get wet, it's time to dry yourself off unless you have a more adhesive surface like a towel to take the water off of you. Surface tension is how difficult it is to break the surface of a liquid, which is why when you go diving into a pool, you don't go spread eagle and land flat on the surface, because the surface tension will slap you, even though it's liquid. Next, property number 2 is the temperature control of water. Water has certain characteristics that lets it control temperature. So it has high specific heat. Specific heat in your chemistry, I remember you guys have calorimetry stuff. It's the amount of heat needed to raise the temperature of 1 gram of your substance by 1 degree Celsius. So a calorie is actually defined by the specific heat of water. Okay, so the high specific heat of water means that you need more energy to raise the temperature of water than most other substances. And couple that with high heat of vaporization, meaning it's the amount of heat needed to evaporate 1 gram of your substance. It means that it takes a lot of energy to turn it into a gas. What that means is that water can take a lot of heat energy from a certain place, and when it evaporates, it does something called evaporative cooling. It takes heat away from a surface. And that's why we sweat whenever our bodies feel like they're overheating. So when the sweat evaporates, the water evaporates, it takes the heat away from the body in the form of the water vapor coming off of you. Next, property number 3 is that ice floats. The reason it floats is because of its stable crystalline structure when it becomes a solid. See, if the water molecules get pushed together very, very close together, it tends to become more stable and crystal-like in its structure because of the polarity of the water molecule. You can't have the positively charged hydrogen ions too close to each other, or the negatively charged oxygen atoms too close to each other. So it forms a regular crystal structure. So what happens is that the ice insulates water that's found underneath it, and the thawing of the ice generates currents. So the cold water goes down while warm water goes up, which is cooled by the ice again. That's why ice is very effective as a cooling agent in our beverages, because the cold beverage will circulate within the glass. Very convenient. And next, property number 4 is the solvent. It's the solvent of life. It's a universal polar solvent. It can dissolve most polar substances, at least over time. So what that means is that it can dissolve ionic and highly polar molecules because of the strong negative and positive charges of its oxygen and hydrogen atoms. So it works against the intermolecular forces of ions and highly polar molecules. Weakly polar molecules just become hydrophilic, meaning they interact with water but they don't get actually dissolved. Take note that dissolution is the separation of ions from each other. So in the case of hydrophilic molecules, they don't get ripped apart from each other. They're just they just have a very high affinity with the water. So water also has power, quote unquote, the power of hydrogen, or basically known as the pH. So without water, there's no such thing as pH. You can note this in your chemical reactions. If you have chemical reactions with strong acids or strong bases, they have to be in aqueous solution. That means they have to be dissolved in water for them to act as acids or bases. So why that is because your water, in the presence of acids and bases, separates into hydronium ions with 3 hydrogen atoms, or hydroxide ions with 1 hydrogen atom, and this has a positive or a negative full ionic charge. And it also manifests the actual pH or power of hydrogen. So the pH level that you see, it's just water being manipulated by your acids or bases so that there's more hydronium ions or hydrogen ions and hydroxide ions. Okay, so high pH means more hydroxide and low pH means more hydronium or hydrogen ions. So in short, water is essential for all forms of life. So what else is essential? What other molecules are necessary? We have the biomolecules. These are molecules that comprise living things, and all of them involve the atom carbon. So in your biomolecules, again, we have your inorganic, your water, and some of your mineral salts, like your actual NaCl, sodium chloride salt, and your organic molecules, which we'll discuss one by one. So organic compounds have a backbone of strong, stable carbon and hydrogen bonds. We'll see that in the structure of each and every one of the biomolecules in a bit. So carbon is the backbone of life because it's the simplest element with the most unpaired valence electrons. If you compare these 4 elements here, these are very simple elements early on in your periodic table, but hydrogen only has a valence of 1, oxygen has a valence of 2, nitrogen with 3, carbon has the most with 4. The only other elements that have a valence of 4 are more complex already and come later in the periodic table. So because of that, carbon skeletons result in high molecular diversity, meaning you have many different shapes. And many different shapes means many different structures, which results in many different functions. So the 4 main biomolecules, we have the carbohydrates, we have the proteins, we have the nucleic acids, and the lipids. So carbohydrates are made of sugars, proteins are made of amino acids, nucleic acids out of nucleotides, and lipids made of many different organic groups. Let's go through each of them one by one, shall we? First, before we go through them, we have the term macromolecules. This refers to 3 out of 4 of them. We have the carbohydrates, the proteins, and the nucleic acids. They're called macromolecules because they can form polymers, meaning many monomeric units, so single units coming together to form very long chains or polymers. So these long chains are formed by dehydration links, which put them together, and hydrolysis breaks them apart. You can see here on the left, the dehydration is called that because hydrogen and the hydroxide on opposite parts of molecules will come together and become water, which is ejected away. So it's like it's losing water. That's why it's called dehydration. And when that water is lost, the 2 molecules are put together into one polymer. On the other hand, hydrolysis is when water breaks the bond between monomers by adding the water and forming a hydrogen and hydroxide end on your monomeric units. So the first biomolecule is your carbohydrates. Its function is as fuel and as a building material. So the monomers, or the building blocks, the fundamental units, are called monosaccharides. These are simple sugars, and the bond between these is called a glycosidic linkage. So in your biochemistry courses, perhaps in college, you're going to discuss that there's different types of glycosidic linkage. There, it can be the same direction or different direction. Okay, so you can see that in your lactose and your maltose. So also take note in your carbohydrates is that your simple carbohydrates are termed as your mono and disaccharides. So mono means one monomeric unit, and disaccharide means two sugar units, or two monosaccharide units. Polysaccharide means that there's many units linked together. So these are your most common examples: your glucose, fructose, galactose, maltose, lactose, sucrose. Sucrose being your table sugar. So these are some of your more interesting carbs. So again, your table sugar, sucrose, is made up of the monomer units glucose and fructose. Lactose, on the other hand, is glucose and galactose, excuse me. So you can see that here on the left. So even table sugar is a disaccharide. Your polysaccharides, we have four main examples. Starch, like in potatoes, is your plant fuel. Glycogen is the fuel of your animals, which is usually stored in the liver. Your cellulose is the plant material, again, in your cell wall. You already discussed that in cellular structures. And chitin is a special protein found in the exoskeleton of most insects and in fungi. Sorry, not protein, it's a carbohydrate. So it's a strong, sturdy material. And this material is also actually the material that's used in your surgical thread, the one that melts on its own. It's made of chitin, harvested from fungi. Fun, right? Next, your second biomolecule is your lipids. So the function of lipids is many, because there are many types of lipids. The definition of lipid is basically any biomolecule that doesn't get dissolved in water. Again, it just doesn't get dissolved in water. So its function is to store energy. It's a material for your membranes. It's hormones. It emulsifies. Again, lots. There's no monomeric unit because they don't form real polymers. And their unifying trait is that they're insoluble in water. Again, that's the defining trait. They're insoluble in water. There's three main types that we'll discuss. We have the triglyceride, the phospholipid, and the steroid. So first type, your triglyceride, or more commonly known as your fats. Therefore, energy storage, insulation, and cushioning. So, the more fat you are, the softer you are. No shame in that. So, the structure is a glycerol base. You can see it in the left here. Glycerol base, that's just the top part, or the leftmost part in this molecular structure. And then three chains of fatty acids. So, these are the chains of fatty acids. And notice they have carbon and hydrogen in very long chains. Again, that makes it highly stable. And each of those bonds between the carbons has energy, a lot of chemical potential energy. And that's why it's an ideal energy storage molecule. But not all fats are equal. Some of them are more healthy than others. So these are some of the different types of fat. So we have in butter and beef saturated fatty acids. These are bad fats. They're solid at room temperature. So your lard, your butter, these are things that can clog your arteries and so on. These are the ones that are associated with a lot of bad press and bad news. But there's an even more unhealthy type, your trans fats, found in stuff like margarine and fast food. So trans fats, these are usually less solid at room temperature, so they don't contribute too much to plaque in your arteries, but they're still linked to heart disease because of their molecular structure. So in your chemistry, a trans isomer means that it's on opposite sides, whereas the cis isomer means that they're on the same side. So your cis unsaturated fats are the healthy or good fats, like olive oil and avocado fat. Okay, so this is healthier because it doesn't contribute to your plaque and it performs the functions that fat is supposed to do. Okay, so these are the three main types of fats found in your diet: your trans fats, your cis fats, and your saturated fats. So lipid type number 2 is your phospholipid found in your cell membrane. So no need to really go through that in detail because we already discussed that in cellular membranes. But its structure is basically the glycerol base, same one as in triglycerides, a phosphate. So instead of a phosphate and two fatty acids, so instead of three fatty acid chains, one fatty acid chain is replaced with a phosphate group, making it amphipathic or partly hydrophilic and partly hydrophobic. Next, your third type of lipid is your steroids. They are a membrane component, and also many of them form your hormones. So your steroids are defined by their structure. There's four rings, four hydrocarbon rings or functional groups. So you can see it here. This is the skeleton of your steroid, A, B, C, and D. Four rings made of carbon. So they're highly stable due to their ring structures and a slight difference in their functional groups, meaning the little groups that come off of the edges here create large functional changes. So some of your examples of steroids, we have different sex hormones. We have your progesterone and your testosterone. We have your menstrual control pills. So you have your estradiol and levonorgestrel, and your immune system suppressants, your cortisol and prednisone. These are also your stress hormones. So again, very much related to how your body regulates itself using hormones. And your third biomolecule is your proteins. They function as cell communications and movement, structure, and catalysts. Their monomer is amino acids, and their bond between them is called a peptide bond. Now, in your proteins, since they're polymers, they have emergent properties coming from their structures. We have a primary structure all the way to a quaternary structure. When we say primary structure, it's just the chain, the normal chain of amino acids. A secondary structure is that chain but folded up into specific shapes. Tertiary structure means that we have a three-dimensional folding pattern already, not just a shape from the chain, it's 3D now. And quaternary means that you have multiple polymers coming together to form a complex protein. So again, we have four levels of protein structure. So your amino acids, your monomers, we have 22, if I'm not mistaken, essential amino acids. So these are amino acids that we have to get from our diet, and they form together to create polypeptides, meaning many peptides or polymer for your amino acids. So what happens is that in your amino acid structure, we have the amino group and the carbonyl group. These are the ones that form a bond together when you have your dehydration reaction. See how there's a hydroxide ion on one side and a hydrogen ion on the other? That's what comes together to form your dehydration reaction. The side chain here, the one that's labeled R, is the one that results in different side chains that make different types of amino acids. So all the shaded ones here are the side chains. Okay? So I won't have you guys memorize your amino acids, but I will probably have you guys identify with a given table. So next, the structure of your protein is related to its function. So again, as we discussed earlier, your four levels of structure result in its function. So at different levels, they have the ability to perform different specific functions. Your primary protein structure, or the amino acid chain, will result in its secondary protein structure, because how it folds is related to how the amino acids are arranged in the uh, polypeptide chain. So it can form an alpha helix, so it can turn into a telephone cord, or a beta pleated sheet, so it's like a folded piece of paper. Okay, so depending on the secondary structure, you can have different 3D tertiary protein structures. So there are some parts that could be a an alpha helix, and other parts that are beta pleated sheets in the same protein. And the quaternary protein structure, again, we have multiple polymers, multiple polypeptides folding with each other. So we keep hearing about folding. So again, structure relates to function. So the proper folding of proteins is essential for its function. So you can see in this GIF here, a chain of amino acids can fold into a specific 3D shape. So since folding is very important, we have special enzymes called chaperonins. These are special structures that help proteins fold quickly and properly. So it's like magic. We don't exactly know how it does that. Your extreme environments, on the other hand, can cause denaturation of proteins, meaning if you cook them in high heat, or if you freeze them in very low heat, or if you have extreme pH levels, you can mess up the folding of your protein and destroy their proper folding. So again, proper folding is essential for how it functions, and denaturation messes up that folding. Your fourth and final biomolecule is your nucleic acids, which is for gene storage and expression, and their bond is your phosphodiester bond. So your nucleic acid monomers are nucleotides which have three parts. We have the phosphate and sugar parts, which form the backbone of your nucleic acids, and the nitrogenous bases, which forms the sequence that can hold the genetic information. So we can see here on the top left your phosphate and your sugar backbone. You can see it in the bottom left here, and then your nitrogenous base just it ekes out on the side to interact with another nitrogenous base on the other side of it with a complementary strand. Your phosphodiester bond is the bond between your sugars and your phosphates to form the backbone. And next, we have your nitrogenous bases. We have two main categories for them. We have your purines and your pyrimidines. We have a mnemonic on the top right for you to remember them. We have the pure silver, which means your purines are your A and G, or your adenine and guanine. Your cut pyramids, your pyrimidines, are your C, U, and T, cytosine, uracil, and thymine. So the general trend is that your nitrogenous bases form hydrogen bonds, meaning they pair up with their complementary pairs across the strands. Generally, a purine will pair with a pyrimidine. In general, it's cytosine and guanine, and adenine with either uracil or thymine. So, adenine has multiple partners. Don't be like adenine. Be like guanine. You good boy. Next, we have DNA and RNA. These are your two main types of your nucleic acids. We have your sugars that differentiate them by name, your deoxyribose in your DNA. This is the sugar here, and your ribose in your RNA. So, based on the name deoxy and ribose, your deoxyribose just has a missing oxygen. If the RNA has an oxygen here, in DNA there's no oxygen over here. Your nitrogenous bases, on the other hand, there's a difference between them. In DNA, there's thymine. In RNA, it's uracil. So adenine pairs with thymine in DNA, and adenine pairs with uracil in RNA. So you'll see that in your sequencing later on. Now, your nucleic acids have what we call a directionality. It's like they have a left and a right side, but of course, molecules flip around in the actual cell. So we don't say left or right. We say the 5' and the 3' end. And these are related to the numberings of your carbon atoms in your sugar base. So the 5' end of your sugar, so this 5' end is usually on a phosphoryl end. It's connected to the phosphate that the sugar is connected to. On the other side, your 3' carbon is connected to your next nucleotide through the phosphodiester bond. Phosphodiester means that the phosphate is in the middle of your two sugar bases. That's why it's diester. Your ester functional group is your two alkyl groups with one thing in the middle, and that thing in the middle is your phosphate. Okay, so again, 5' end and 3' end, so you can just think of it as left or right. So they are always anti-parallel, your complementary strands. So that means that if one strand is going from 3' to 5' in one side, your other end will be from 5' to 3'. Okay, so again, they go in opposite directions, anti-parallel. Okay, so what happens to these biomolecules? What processes occur to them? We have your chemical reactions, and in the biological context, one of the main ones is your redox reactions. But let's clear up some of the terms first. So substances have categories. Your pure substances can be called your elements or your compounds. Your atoms are single elements. So single atoms of your elements, the fundamental unit of matter, supposedly. And your elemental molecules are your covalently bonded elemental pairs. So like oxygen gas or hydrogen gas. Your molecules are your covalently bonded compounds. So water is a molecule. When we say molecule, you should know already that it's covalently bonded. And your ions are your, of course, ionically bonded compounds, like your sodium chloride. So there's many types of bonds. Covalent bonds share the electrons, ionic bonds transfer them, and hydrogen bonds, they're not a bond between atoms, but bonds between molecules. And they're purely electrostatic, meaning they're just attracted because of opposite charges. So again, hydrogen bonds are strong, but they're not between atoms. They're between molecules. And of course, differentiating important reactions, dehydration, put stuff together through oxygen usually, or sorry, uh, covalently bond monomers together through oxygen or water. So you can see it here. There's oxygen being oxygen's like holding the hands of the two monomers in this reaction here. You can see that your OH on opposite sides becomes a link with the O. So you'll see that in glycosidic linkages that oxygen holds them together. Your hydrolysis reactions break bonds by adding a hydroxyl and hydrogen from water. So you can see it here, your OH and your H that are between the two, keeping them apart. And finally, an important type of reaction in the biological context is your redox or reduction oxidation reactions. These involve a transfer of electrons, but it doesn't always result in ionic compounds. See, we talked about ionic bonds earlier, that they form due to the transfer of electrons. In redox reactions, it doesn't always form a bond between different atoms. Okay? It could just be a transfer of the electrons or basically a rearrangement of electron density. So what do we mean by that? So let's take a closer look at redox reactions. The basic redox reaction you can see in this diagram here is that oxidation means there's a loss of electrons and reduction means a gain of electrons. So in this case, A lost electrons became positive and B gained electrons becoming negative. So again, that's basically it, a transfer of electrons. So complete electron transfers, we can see it through the charges. However, in the biological context, it's usually a change in electron density. So you can see it in this bottom left one. There's most of the electrons found in the bonds of your carbon and hydrogen here in the reactants in your C4H10. After the reaction, the electron density was switched to have more electrons between oxygen and carbon. Basically, oxygen hogging the electrons, more or less. So, your redox reactions can again be a partial transfer of electrons. So instead of an equal sharing of electrons resulting in low electrostatic charges, it can have a rearrangement so that more electronegative atoms like oxygen can end up hogging the electrons themselves. So gaining an oxygen atom in your reaction or losing hydrogen atoms usually means oxidation. Usually means that that molecule was oxidized. That's why it's called oxidation, because of the addition of an oxygen that hogs the electrons. Because it hogged the electrons, the molecule itself lost electrons. On the other hand, losing an oxygen atom or gaining more hydrogen atoms means reduction most of the time, because again, without that hogging electronegative oxygen atom, you have more electrons to go around and be shared in the molecule. So it's like a gain of electrons or reduction. Okay, so again, don't forget, in the biological context, the gaining and the losing of oxygen atoms is key to your oxidation and reduction. So redox reactions are extremely common and very important in biology because they transfer energy. The less polar bonds, like those between your carbon-carbon bonds, are stable and have higher potential energy. So oxidation can help release the energy from those bonds. Because of the transfer of electrons, energy is released, and electrons can carry that energy and transfer it to other molecules. We'll see that later in your metabolic processes. Just remember that your redox reactions are key to transferring that energy in the bonds. So chemistry is practical. It has applications to life, to biology, to understanding what keeps us alive. And the question is, what reactions, what interactions keep you alive? What keeps you stable? What gives you energy in your life? These are the things that we should understand and give importance to. So that's pretty much it for this session, and I'll see you guys next time.
Party over.